Lesson No. 15 Genetic Engineering of Plants. Genetic engineering, the science of adding, removing or replacing genetic units in order to achieve permanent and heritable changes in plants and animals for the benefit of mankind. Terms such as gene surgery, gene therapy, gene manipulation, gene transplantation, etc. have been synonymously used with genetic engineering. Genetic engineering refers to techniques that are used to manipulate, move, recombine and propagate DNA. Changes nature of plants, animals and microorganisms happens from the beginning of life on Earth. Recent times scientists developed techniques to modify living things on molecular, genetic level. Significant outcomes of these genetic modifications include increased food production, reliability, and yields, enhanced taste and nutritional value, decreased losses due to various biotic and abiotic stresses, such as fungal and bacterial pathogens. Genetic engineering developed due to the work of many researchers in various fields of biochemistry and molecular genetics. There was even an assumption that genes are protein in nature. Only in 1944 Avery, MacLeod and McCarty showed that DNA is the carrier of hereditary information. In 1953, J. Watson and F. Crick created a double-stranded DNA model. Hargobin Khorana, Robert W. Holley and Michelle Nirenberg in 1960s developed the concept of the genetic code. In 1960s, the properties of the genetic code was clarified, and by the end of the 1960s its universality was confirmed experimentally. The first recombinant DNA molecule was made by Paul Berg in 1972, by combining DNA from the monkey virus SV40 with the lambda virus. The first GMOs were bacteria in 1973. Coli, DNA cloning method was developed by G. Collins and B. Coleman in 1977. Important advances in creating recombinant DNA included the discovery of enzymes. Enzymes used in the construction of recombinant DNA divided into several groups. Enzymes by which DNA fragments, restrictase enzymes. Enzymes that synthesize DNA on a DNA matrix, polymerase, or RNA, reverse transcriptase, enzymes connecting DNA fragments, ligases, enzymes that allow for a change in the structure of the ends of DNA fragments. Significant enzymes, restriction enzymes and DNA ligases, and the development of polymerase chain reaction and sequencing. Main steps of RNA creating recombinant DNA technology. Genetic engineers must first choose what gene they wish to insert, modify, or delete. The gene must then be isolated and incorporated, along with other genetic elements, into a suitable vector. This vector is then used to insert the gene into the host genome, creating a transgenic or edited organism. Genetic engineer will, at different times, prepare 3 different types of DNA: total cell DNA, genomic DNA, plasmid DNA, phage DNA, if phage cloning vector will use. Methods of genetic engineering. Vectors in RNA technology. Microbial vectors used to transfer and stably integrate specific new genetic material into the cells of target plant species DNA. Agrobacterium tumefaciens is a naturally occurring soil microbe best known for causing crown gall disease on susceptible plant species. The Biolistic method, microprojectile bombardment, naked DNA delivered to plant cells by shooting them with microscopic pellets to which DNA had been adhered. Electroporation, supplying known DNA to the protoplast culture medium and then applying the electrical pulse temporarily destabilizes the cell membrane, allowing the DNA to enter the cell. Microinjection DNA can be injected directly into anchored cells. Some proportion of these cells will survive and integrate the injected DNA. Application of genetic engineering in the field of agriculture. 1. Creation of resistance varieties of plants. Various novel plants with resistance to various diseases, pests, and stresses have been created using recombinant DNA technique. Genes 2. Biofertilization. Molecular nitrogen in the atmosphere is converted into biologically usable form by nitrogen-fixing microorganisms, for example rhizobium. NEF gene from bacteria. 3. Increase the protein content. Proteins contained in seeds of many plant species, deficient in some proteins. French bean protein, phaseoline, has been expressed in sunflower. Modern uses of genetic engineering, finding out where genes are expressed in the body. Manufacturing insulin. Crops which produce a greater yield. Crops resistant to disease. Possible dangers of genetic engineering. Any new scientific discoveries offer the possibility of both beneficial and destructive effects. Some of the possible dangers, due to manipulation of genes, might, by accident, result in the origin of various new kinds of diseases or organisms containing fatal genetic element. There is a risk of creation of drug resistance germs and outbreak diseases against which there is no known prevention. Possible dangers of genetic engineering. Any accidental escape of laboratory strains may create havoc on Earth. It may contaminate a large population. Introduction of gene like that of viral cancer into bacteria through plasmids may involve the risk of introducing these harmful genes into man when these bacteria infect human. Hybrid genomes may create some serious ecological problems, the nature of which is still unknown. Naturally, all these harmful hybrids possibly raise many moral, ethical and legal questions. Modern methods. Genome editing is a type of genetic engineering. DNA is inserted, replaced, or removed from a genome using artificially engineered nucleases, or molecular scissors. Silencing the gene of interest by short RNA interference, siRNA. Genome editing with nucleases such as ZFN. Genome editing was chosen by Nature Methods as the 2011 method of the year. Thanks for your attention.